Hey guys, hope y'all had a great weekend. Today we'll be learning about DNA cycle sequencing, its purpose, and the steps it takes to do it. To start off, we need to know the materials that are needed to complete DNA cycle sequencing. First, the first thing that needs to be done is plain and simple, just a template DNA, which is a strand of DNA that you want to sequence. Next is a DNA primer, which is kind of a short version of the DNA template, but it's the complementary version. So if it has an AGG, the primer would have TCC. And this will connect to the DNA template and prime and get it ready to start sequencing. Next is the DNA polymerase, which is an enzyme that binds with the template, which also prepares the template for nucleotides for it to start sequencing. And that's what's next is nucleotides, which we should have learned in freshman year biology, which is adenine, cytosine, guanine, and thymine. In in this in DNA cycle sequencing, each of the nucleotide bases have a color coordination. So adenine is green, cytosine is blue, guanine is yellow, and thymine is red. Not only do they have nucleotides, but they also have DD and TPS, which are deoxynucleotides, which are sort of the same as nucleotides. They will be put into sequencing the same way, but they have a different chemical composition, which we will explain later in the video. Next is a thermocycler. This is where the first part of DNA cycle sequencing is performed when the DNA fragments are broken up. This is what helps break apart the DNA fragments and create the lab environment. Next is a polycromate gel, which after the fragments are broken up and sequenced, this will be put in this gel to sort out the DNA fragments in different lengths, and then it will be put into a DNA sequencer, which is the last material that is needed. Next, we have the steps. The first step of DNA cycle sequencing is putting every material into the thermocycler, which you put just like you would put all the ingredients to make a cake. And then next, the thermocycler is heated to 96 degrees Celsius. This heat is what's going to help break apart the DNA fragment into two strands, which is the first step needed to start DNA cycling. Next, like in the steps above, there's a primer added, which the primer is the complementary strand to the sequence of the DNA template. And to perform this, the thermocycler is dropped to a temperature of 50 degrees Celsius, which allows the primer to bind with the DNA template. Next is the DNA polymerase, and this binds also with the template along with the primer, which will allow the nucleotides and sequencing to start. And to do this, the thermocycler is brought back up to 60 degrees. The temperatures are very important here because as the first step, 96 degrees heat is what's going to break the hydrogen bonds down in the DNA strands, and to lower it, the primer only works at its best at certain temperatures. So if it's too high of a temperature, it will be denatured along with the DNA polymerase, which is an enzyme, which an enzyme's a protein, and proteins function best at certain temperatures, and if it gets too hot, they will become denatured and not work. So 60 degrees Celsius is one of the best temperatures for DNA cycle sequencing. And the fourth step is to add nucleotides in DDTPS, which is added to the template like regular sequencing would. So if the template is AAGGCC, the nucleotides will start filling in TTCCGG, or the opposite or the complementary of what the template is. So they will start throwing those in randomly that will line up with the DNA template complementary, but there is one difference. There is also the deoxynucleotides. So when one of those are added, there's a special reaction that happens where they know once the DDTPS is added into the strand, the sequencing will stop and will continue on another strand of DNA that was put in the thermocycler. And this is what creates the many lengths of DNA fragments, which will soon be sorted out. Next, it is put into polyacrylamide gel, which will kind of sort out the fragments from the smallest to largest. And this is important because for the next step, it is going to be put in a DNA sequencer for electrophoresis and analysis. And the way this works, when the DNA fragments are put in, there's a laser that reads each of the DDTPS, the deoxynucleotides, will read each of the ending fragments, which will put together the sequence for the DNA, and this is read by a laser, and once the nucleotides hit the laser, they glow, so they show like the base that they are, like red would be thymine, green would be adenine, so they'll know, since by the way that they glow, which order the sequence is, and it starts from the smallest fragment to the largest fragment. And after the DNA sequencer figures out the order of nucleotides, it is put into a graph of colored peaks, which will show the way the nucleotides are organized in order in a sequence, hence the name DNA sequencing. So there will be a yellow peak, a green peak, a blue peak, a red peak, and you will find, depending on what color the nucleotide bases are colored, will show how the DNA is sequenced. So you will take that graph and then figure out what the sequence is. I know since we just learned about PCR polymer chain reaction, there may be some questions of how these are similar and different. This is a very common question, and there are some similarities and differences. For example, PCR, regarding to nucleotides, PCR has only D and TPS, which are just base nucleotides in the sequence. But cycle sequencing has the regular nucleotides and the deoxynucleotides, which both perform different functions in the cycle sequencing. Next is primers. There's a different number of primers in PCR and cycle sequencing. In PCR, there are 2 primers because it will not synchronize. It will sequence both DNA fragments when they are broken up and begin to copy them. But in cycle sequencing, it only starts to sequence one because there will be an abundance of fragments to use, and they only need a certain amount. And the length of PCR is different to cycle sequencing because the primer starts at one end and the polymer continues to the strand ends. But with cycle sequencing, primers start at one end and the polymerase continues the sequence until it reaches a DD in TPS, and then it stops. Next is the purpose of each of them. The PCR, it creates multiple copies of a single strand of DNA, which can be important in testing and figuring out illnesses and diseases. In cycle sequencing, the purpose of this, it determines the sequence of nucleotides in a DNA strand, which can help you pinpoint pathogens and other mutations in genes. And lastly, the difference between the splitting of the DNA in PCR and cycle sequencing is the helicase enzyme breaks down hydrogen bonds to separate it in PCR, which is different from cycle sequencing because in cycle sequencing, heat causes hydrogen bonds to break or be denatured, which will cause the split in the DNA fragments, which is used to start the process of cycle sequencing. I know this may be a little confusing at first, but once you get the hang of it and perform the task, which will be in one of our next assignments, this will show you how they are very different, but they can be worked together and how they can help doctors and scientists figure out more about DNA and how to perform in the human body and just some more information we need to know. So check modules and get the homework completed by next week. If you have any questions, just email me, and I hope you all have a good day. Bye!